work. Dr. Susan Abbatiello has a long-standing experience in mass spectrometry. She's a bioanalytical chemist with experience in biomarker candidate discovery and verification using proteomics techniques such as, of course, mass spectrometry, including isotope dilution mass spectrometry, multidimensional liquid chromatography, and high-field asymmetric waveform ion mobility spectrometry. Her work now is focused on method development and studies of reproducibility and robustness of SRM-MS, quantitation of peptides and related proteins from digestive plasma and cell lysates. And today she is going to talk about a long-standing and very tricky issue that is going from the discovery phase in proteomics to the targeted phase in proteomics and later on ending in biomarker discovery. Susan, it's a real pleasure to have you here with us today, and it's all yours. Now is focused on method development and studies of reproducibility and robustness of SRM-MS.
Great, thank you. All right, thank you for the opportunity to join you here. It's always fun to, especially now, be able to interact with people without having to necessarily get on a plane. And so I'm happy to share with you my experiences in moving from discovery to targeted proteomics using LCMS, and a lot of the little tips and tricks I've picked up along the way to help avoid problems. You can't completely not have problems, but at least we try to avoid them. All right, so just to give you a little background of who I am and how I've collected my excuse me, my experiences along the way. I received a bachelor's in arts and chemistry from the College of the Holy Cross up here in Massachusetts in the US. And right after I received my bachelor's, I did not want to go to graduate school. I do better in the lab rather than the classroom. And so I decided to get a job as a research associate at a company called Genetics Institute, which manufactured protein biotherapeutics. And there I did a lot of LC-UV based work and became interested in mass spectrometry, but I wasn't allowed to use it until I had a graduate degree. So from there, I went to the University of Florida and received my PhD in analytical chemistry, focusing on mass spectrometry, in particular, quantitating a drug-resistant marker in acute lymphoblastic leukemia. And then not entirely sure what I wanted to do, I went to the University of Pittsburgh, where I did a postdoctoral fellowship at the Hillman Cancer Center for a little bit before returning back to the Massachusetts area to the Broad Institute, where I worked for Steve Carr and the proteomics platform as a research scientist. And a lot of the work there was focused on targeted biomarker detection. I've had a long-standing interest in FAMES, ion mobility, and so I decided to take a break from doing research, biomarker research, and I worked at Thermo for a while, developing the FAMES hardware that came out a few years ago, FAMES Pro. And then more recently, 3 years ago, I joined Northeastern to run the mass spectrometry core facility at the Barnett Institute. And so picked up a lot of experiences with small molecules and polymers and synthesis products. And it was a really exciting couple of years. But just about 2 months ago, I became a principal consulting scientist at Waters Corporation. So I'm back in the research for the vendors, helping to improve the technology. So you can see I've kind of done a lot of little things along the way. And I have a lot of experiences about that to share with you. So looking at application of liquid chromatography mass spectrometry for discovery of biomarkers, we have this usual paradigm where we begin with, if we're looking at a discovery type experiment, what is changing in our sample? And we need to generate data in order to see changes across different biological replicates or different simulated samples. And these samples can be things like plasma, tissue, proximal fluids, or you can even grow cells in culture and monitor them that way. And then we do this discovery step where we look at tens of samples and as many analytes as we can possibly detect using LCMS, usually high resolution, high mass accuracy instruments. Then once we look at the data, we can begin to narrow down what is actually changing in these samples and to what extent they're changing, and the variability of those. In order to do that, we need to increase the number of samples we're monitoring and decrease the number of analytes. And this can proceed in a semi-targeted way using triple quadrupoles or high resolution accurate mass instruments. And this is a verification step, which is normally done in blood in a larger population. Here we're moving to blood because it's easier to obtain than tissue or proximal fluids, with the end goal that eventually we would be able to develop a clinical based method, something for diagnostic purposes, where we could analyze thousands of samples and look at just a small number of analytes, maybe a panel of peptides or proteins, and we would be able to do this from a blood sample with a large population. This verification step sort of begins now with the hypothesis-driven how much is changing. So sometimes the information feeds in from discovery proteomics experiments. Sometimes it feeds in from the literature. Sometimes it feeds in from interesting biology, where you already have an idea, perhaps from a Western blot or an ELISA-based assay, that there is a protein changing, and you may not have to do this larger scale discovery-driven step. You can begin right with proposing a hypothesis of certain proteins that are changing. And from that, it really follows the same workflow. So for discovery proteomics and trying to determine accurately what is changing in the samples, there are a couple different approaches, and you can kind of weigh the pros and cons of each that are best suited to your situation. So the simplest and probably least expensive from a reagent standpoint is label-free quantitation. In this case, you've got 2 or more samples where you want to evaluate changes in the proteome across these samples. And so you digest the sample from proteins into peptides, analyze them, and then look at the signal changes based on the accurate mass, retention time, protein and peptide identifications that are made. In order for this to have meaningful data, you usually have to generate several replicates for each sample. So usually 3, a statistician would ask that you do more. And this tends to, then you just look at the signal of the MS between the 2 samples to see what the change is. And so it takes into account the variability within a sample and then the biological difference across samples. This can be time consuming because you have to run each sample many times to generate the statistics to show what is indeed changing. And this approach is less precise at peptides that are of lower abundance. A next type of quantitation, relative quantitation approach is chemical labeling. This is something you can do with, for example, isobaric labeling like TNT or iTrack. And this provides differentiation of what can be 2 samples or up to 15 samples that are combined in the workflow process after digestion. They're labeled, the peptides are labeled, and then the samples are all combined. And so all of a sudden, analysis of 15 samples can collapse into 1 chromatographic run. And there are reporter ions that allow you to quantify the differences either in the MS2 or in MS1, depending on what type of label you use. Often because the samples are so heterogeneous and complex, there is something called compression, where if you have large changes in protein concentration between 2 samples, they may be underestimated, meaning a change of 10-fold might actually look like a change of 8-fold. So it tends to be less accurate at the higher concentration differences, but this can be overcome by doing fractionation. But now if you do fractionation, you're taking your 15 samples, combining it into 1 run, but actually fractionating that further to maybe have back out to 15 runs. It is better precision-wise than label-free, but it does come at the cost of the chemical reagents. And then finally, metabolic labeling. This is probably one of the best approaches for doing robust relative quantitative methods, because this incorporates a label right up in the cell culture. And the samples, as soon as they are labeled and grown to the point at which they need to be harvested, they are combined because they're already labeled, and state A and state B are differentiated by their isotopic isotopically labeled lysine and arginine. And then anything that happens in the sample workflow process happens to both sample types at the same time to the same extent, theoretically. And then you look at the changes in the MS signal to quantify differences in the peptides. Now, this is a bit difficult because you're limited with the number of states that you can combine. I think right now it's up to 3. And it's more difficult to label living organisms. You can do this in cell culture, but you can't take somebody's plasma that's already been removed and SILAC label them. So this has its limitations. And of course, as I mentioned, the precision of the quantitation is increasing as you move from label-free to chemical labeling all the way to metabolic labeling. There have been studies of SILAC labeling with mice, and there was a paper that came out many years ago, but it's not something that's routinely done. So the typical discovery to targeted workflow that we'll be going over here starts, you have a large number of samples or even a small number of samples, and you analyze these by LCMS, usually after tryptic digestion. And then you search these data against a proteomic database and you identify the peptides and proteins that are present in the sample. And then you can look for changes between the two. And there are a lot of different software packages that are made by vendors or made open source that you can use. And then from that list, you can sort of narrow down everything to the protein target that you want to select for accurately quantifying. And so now you're going from looking at everything in the sample to really narrowing it down to looking at a smaller portion of what is in the sample. Now, I will say I've used the software Skyline for a long time, and so it is integral in my process. It's free software from the University of Washington. If you Google Skyline and proteomics, you'll find this. You can download it, and it's a very powerful suite of software that works for peptides and small molecules. And so I'll be highlighting some of the features for it here because it's just a great software package and has a lot of things for the tips and tricks that I'll be mentioning. So in Skyline, I take the searched data and I'll generate spectral libraries that I can visualize as I click on a particular peptide. I can add my peptide sequences or even add a protein, and then the software will in silico digest and give my peptides to me with their predicted mass to charge and product ions. And then I go through and I often reload my discovery data into Skyline, and I actually look at the peaks. Now, this may be a long and labor-intensive process. There are some shortcuts you can make that I'll go over, but this is generally what I like to do as I'm moving from a discovery to a targeted method. And this is an iterative process. You don't have to do everything, but if you do, you have the best chance for a really robust assay that will minimize downstream data analysis issues. So as an example, this is from an ASMS poster a few years ago. We wanted to look at plasma proteins and see how many we could detect on a triple quad in one run. And we were able to target 241 plasma proteins and 950 plasma peptides. And this was just trying to see how fast the instrument was. There was no biology behind it, but it was something that we were able to do in just a couple of days. Okay, so first, when we're looking at moving from discovery to targeted or just winding up with a targeted proteomics workflow or quantitative workflow, it's really important to stop and understand your project. And there are some things, I have little check marks that you can look up in the literature. And I will mention, this is super important. And I've been burned by having access to a lab and just saying, well, let's just go in and run the experiment. And then we find out that the particular things we're trying to target, we would need to do a protein pull down and an anti-peptide antibody pull down to be able to get enough material to see them by LCMS. And we were just trying LCMS. And so in cases like that, you can prevent yourself going down the path for failure by just asking these questions. Can you look up what the protein concentration might be in the sample matrix? Has it been studied before? Are there other biological techniques that are reported in the literature? Another is, how complex is the sample matrix, and can you make it a little less complex? Can you remove some things that might interfere or cause column loading issues? You can also look, particularly as we progress in time, people publish spectral libraries, people publish raw data. You can see if the empirical data exists for your peptides of interest and see how they look, and see, and chances are if they don't, you may have challenges ahead. And so just keeping that in mind. And then, is the linear range of detection already known? This is a good question to ask, just understanding if you're targeting a protein that tends to change, does it make big changes? Does it make small changes? And can you get the precision of your method to meet those needs? And then these other questions, I'm not going to go through them all, but these are ones where when I sit down, particularly if I'm supporting the work of a collaborator and I'm doing the LCMS and they don't have one, these are all the types of questions I have, particularly around internal standards. Is enrichment of the sample needed? How much funding is available to support the work, and so on. So this picture is an example from a paper that was published a few years ago, where we had some stimulus support from the US government to try to, this was a high risk, high reward project where we were trying to evaluate what the effect of ER and HER2 status was on the proteome in breast cancer cell line cultures and in some tumor tissues. So we collaborated, this is the work I was doing at the Broad Institute and Steve Carr's group. We collaborated with Mandy Palovich at the Fred Hutch Cancer Research Center and with a lab in Seoul, South Korea. So we had 3 different analytical labs, and we began by working with 4 different cell ER, HER2 cell statuses. And you can see we've got ER positive, HER2 positive, and then the positive and negative switch until they're both negative. We also had some non-tumor normal tissue to compare. So initially for the discovery approach, we had all these individual cell samples, but we pooled them into their cell status so that we could do the discovery process and not run them all individually. And so from that, we took it through a data processing step. Let me show you, where we had all of these cell lines with the different status, a total of 30 of them, but they were pooled into these 4 individual subtypes. And then we ran them by LC, high resolution accurate mass spectrometry, and generated a whole data repository where we began looking at proteins and peptides that were changing. And the goal was to pick the top 300 proteins out of everything that had changes between these different subtypes, especially and compared to the non-tumor, and then set up targeted methods for 600 peptides. So this means 300 proteins, 2 peptides per protein, and then we also included label standards. And so there are lots of different ways you can approach the data analysis and understanding what is changing in your sample. And one of the approaches we took was to look at the cellular compartments defined by Go, and the proteins are shown in columns, and their cellular compartments where they're located are in rows. And we just look for things that are changing. We also look at biological processes defined by Go and the proteins in these columns, and then see how they are grouping by subtype, and if they are subtype specific, whether their change is specific. It was difficult to come up easily with 300 proteins that you could look at the data or a heat map or clustering analysis and say, oh yeah, there's definitely 300. So we did have to extend it to some that maybe weren't changing as clearly, but the goal really was to sort of push the throughput capacity and the ability to run a lot of peptides in one analysis. So the way we broke this down, let me go back one slide. The samples were all prepared at one central site. And so the peptides were digested, desalted. They had isotopically labeled peptides for all 600 peptides present. And then they were distributed into 3 groups for each of our 3 sites. And in order for us to carry out the analysis, we split. So there were 3 plates, and then each lab ran 2 different LCMS assays. One was a site-specific one that was 75 proteins, 150 peptides at one particular site that was not repeated at the other site. And then the interlaboratory LCMS method was one that all 3 labs ran on the same samples to prove that we were getting reproducible and accurate results. And this process took several months. I think it obviously takes longer when you're working with multiple external collaborators. But all in all, we were able to pull together a method that was using nanoflow chromatography and triple quadrupole scheduled multiple reaction monitoring with an 8-point curve. And then we reanalyzed those cell subtypes as the individual breast cancer cell lines to see what the actual changes from individual cell lines were. These were all done with process replicates in preparing the samples and at 2 different cell lysate protein concentrations. So as I mentioned, we had these 3 labs involved, and every lab ran these 2 different methods in triplicate and on duplicate cell lysate dilutions. And this is something that at the end of the day, we were finding good reproducibility. I don't have a lot of great figures to show for this because it really was more of a statistical analysis. And then looking at biologically what proteins together individually seems to contribute to the HER2 status in these breast cancer cell lines. But this was a basic way of going from discovery to targeted. Now, when moving into a targeted method, these are, again, there's some more questions that I think it's worth asking, like, why was this protein selected? Is there empirical evidence? Is there something in the literature? Is there something biologically significant that supports perhaps what is happening in the cell or the sample or the individual? And then how much protein can you collect from a sample? Do you have to do enrichment? Is it something where the protein you're targeting is so low abundant that you can't load enough of your sample on your column? Then this could be an issue. How many targets? I don't think this is as big a deal. The instruments these days are so quick that they can really target lots and lots of peptides. In the olden days, when people did this on triple quads, there used to be a target limit of maybe 10 or 100 peptides or a number of transitions. But I think the technology has evolved enough that this is less of a concern. And then the cost, is there enough to support the research, especially in academia? This is super important because you want to make sure you have enough money for the project and for your grad students or postdocs and all of the supplies and materials. So of note, internal standard peptides for targeted workflows cost about $500 for preparing them and having a good amount of stock, and they tend to last, but it is a large upfront investment. And then there's the time it takes to do the method development on the LCMS portion. And then the hardware itself, if you don't have these instruments, do you buy them? Do you collaborate with someone else and have to help support their maintenance and upkeep? And then of course there's time. The time not only it takes to analyze the sample on the instrument, but also the data analysis time. It is not fully and perfectly streamlined automatic. In fact, I encourage people to go in and look at their data. And so that does take a little in-person time. And then biological interferences are, before you start, you want to make sure that there's no other protein that has the similar sequences as yours or potential for interference that may confound results. And what would be the potential for false positives. And so again, like I had mentioned, never underestimate the literature. And here are a few questions and steps that you can ask yourself. You know, one of my favorites is if you need to get a liter of blood in order to detect a protein from an individual, that's a big ask. I think that wouldn't work out so well. And so you need to make sure that your analyte is present and reachable by the LCMS technologies that we have. And then this is just, I think, a great saying. A month in the lab can often save you an hour in the library. And this is exactly what a lot of people do. Well, I have a lab, I can just go in and figure it out. But sometimes if you go into the library and do a quick analysis, you'll find out maybe a better approach than the original one you had planned for. And then finally, sometimes when you go and look into the literature, you might find nothing and you might say, oh my gosh, if I could do this experiment, it'll be so easy to publish. And that is sometimes, not most of the time, it's not true, because sometimes the things you're looking for that haven't been published, they haven't been published not because they haven't been thought of or tested, but because there were no positive results. And we often can't publish confusing things or things that don't have positive results. And so they go unreported and often repeated by researchers unsuccessfully. So one of the main challenges we have looking at biomarkers is if you think about where you would want to analyze a biomarker from, what kind of sample you want to collect, blood and plasma is one of the simplest. And it also is great because it contains proteins that are shedding from cells, and blood goes everywhere in your body, unlike things like saliva or urine. So it's a great way to maybe assess what's going on in the human system. However, the protein concentration range is so large. You can see the picogram per milliliter concentration goes from log-based 10, so 10 e to the 11 down to super low concentrations. Now, what we see in blue, we can detect by conventional LCMS, but to get the greens and the reds, these are at low enough concentrations that we do need to do some kind of enrichment step. And so that has contributed to the basic workflow of using potential for protein level enrichment and peptide level enrichment. And so here I'm just showing an example where you start with a protein sample that you either get from cell lysate, tissue, plasma, so on. And your usual input might be 10 to 100 microliters of plasma or 10 to the 7 cells, particularly for a discovery experiment. And then at this next point, you can do a protein level enrichment by either amino affinity, by removing the most abundant proteins from a sample and retaining what is left over. You could do cellular fractionation. You could do in gel digestion and just cut out the band that you think is your protein and get rid of everything else. So there are a lot of different options for protein level enrichment. For peptide level, you can do IMAC, which enriches for phosphorylated peptides. You could do an offline separation, such as basic reverse phase, where the pH is high, instead of acidic reverse phase, where the pH is low, or strong cation exchange. And here you would separate out a series of fractions that are separated in a way that is different to acidic reverse phase. So if you collect your fractions across your gradient and you inject one that came off early in either basic reverse phase or cation exchange, it would still have a bunch of peptides that separate over the entire retention time in the acidic reverse phase that you do online. This, however, increases the amount of time you have to do for data acquisition. So nanochromatography has been super helpful because it improves the sensitivity of detection because the flow rates are so low. And so this has been a pivotal point in this workflow process over the years, and NanoFlow has become a lot easier to use with companies focusing on making it work better, detecting leaks before you actually have leaks, because as we know with nano, the flow rate is so low, it often takes time to know if there's leaking going on. And then historically, most often a triple quad has been used where you can separate out peptides based on their intact precursor and their product ion signals. But now, due to the high resolution accurate mass instruments out there, those instruments are also used. So this just gives you an idea of normally what we're able to detect on average, and this is a little hand wavy, but if you load about a microgram of digested protein that has not been enriched onto a column for LCMS analysis, you can usually detect hundreds of attomoles or lower of a particular peptide. If you do an IMAC enrichment for phosphorylated peptides, now you're removing all of the non-phosphorylated peptides and most of the sample. So when you load a microgram of sample, you're loading lots and lots of phosphopeptides. And so what that translates to is hundreds of attimoles per 1 milligram of protein. And this is also true for anti-peptide antibody capture. And this just shows you the sample equivalent volume. If we're talking about plasma, because it's so concentrated, it's about 60 milligrams per milliliter protein concentration. So if you want to load 1 microgram of plasma after digestion, that equates to only about 15 nanoliters of that plasma sample without any enrichment. If you do a plasma depletion where you take and remove the top 14 most abundant proteins, now the sample that's left to get about a microgram, you would be loading about 220 nanoliters. And you can see as these enrichments proceed sequentially, you wind up loading a large sample equivalent, which is how the enrichment all works. So NanoFlow, as I mentioned, improved the sensitivity, and this just shows on the y-axis the sensitivity improvement moving from a 2.1 millimeter scale column all the way down here at the right, where the flow rate is just under a milliliter per minute. And as you reduce the column inner diameter and reduce the flow rate down, sure, your chromatographic separation time increases, but the sensitivity factor is now 50-fold compared to where it started at 2.1. So if you're not looking at super low abundant analytes, you could go into this intermediate range where you take a little bit of hit on the sensitivity, but you can go at a faster flow rate for higher throughput. And so these are just options to think about in the trade-off process. There's also your LC gradient. So this is just an example where we were trying to run a gradient and improve it based on this green one, where we had a lot of peptides coming out early on. Unfortunately, due to the NanoFlow setup, this equilibration amount of time was almost 100% necessary, which is frustrating with NanoFlow. But we began looking at these different gradient options because you can change the gradient. You don't always have to use something that was published or something that was just established in the lab before you joined. So this is the reason why. This one gradient shown here had been working very well for a method, but when we decided to swap to a different set of peptides and use it on another project, we noticed that these are just the peptide standards. They're all coming out in this little less than 10-minute region and all packed together. And we had to remember that it wouldn't just be the peptides that we want to monitor that are coming out here, but everything in the sample would be coming out here because there's really nothing coming out later on. And so by adjusting the gradient, you can see these peptides are migrating later and later. Their peak shape seems to be fine. You know, we wanted to make sure we weren't losing peak intensity, but we also were struggling with the peaks being wide enough at the front end of this gradient. And so kind of spreading everything out a little bit seems to help. And if we look at this in a mathematical sense, and we want to know what the duty cycle is of the instrument with regards to the chromatographic duty cycle, it's very low. We were at 11 minutes over a 90-minute separation. We were at 12% duty cycle meaning the amount of time the instrument's actually doing something versus the whole assay time. And as we spread these things out, we were able to get up to about 38% duty cycle, which is more in line with where we would like to be. With chromatography, it's really difficult to get to 100% duty cycle unless you have columns running in parallel. So moving from discovery to targeted, do you simply rely on the empirical data to pick your peptides, or do you throw additional ones in that may not be detected, or do you have any choices? Do your peptides pick you and you just go with it? So some things to think about for picking your peptides is what is preferred, obviously, is a unique sequence that is unique to the protein you're targeting. And then the 6 to 25 amino acids in length is just, it's conventional because anything smaller than 6, for 2 reasons. Often it will only be singly charged. And also if it's not, its mass to charge comes in and is detected usually below 400 mass to charge. And that tends to be a very noisy region in the mass spectrum. And so 6 to 25 is better. 25 or larger tend to be quite large and can be quadruply charged. That leads to multiply charged product ions, which just can kind of add difficulty in the data analysis. I always recommend 2 or more proteins per peptides per protein. And then it also helps if there's one or more basic residue in the peptide. For example, if you're not using trypsin, because trypsin cleaves on the C-terminal side of lysine and arginine are basic. So tryptic peptides always have the N-terminus and the C-terminus providing charges. But there are other amino acids that can do that. So if you're looking at the C-terminal peptide of a protein, or if you're looking at endogenous peptides, having at least one or more basic residues will help with charging it up. And then it's nice if it produces lots of product ions. If you fragment a peptide and it gives you 2 product ions, that's not very informative. And it will help with, it will hinder identification. Recommendations on things to avoid are post-translational modifications if they're not important to your project, because they may be occupied or not. Multi-basic stretches like lysine, arginine sequences in the protein can often produce what we call ragged ends. And that just means the peptide may appear in several forms where it's fully cleaved, fully tryptic, or it has an additional lysine or arginine on the N or C terminus. And so this is useful to know because you might have to begin monitoring for multiple forms of your peptide if you're locked into that particular peptide sequence. Methionine is well known to oxidize, and that will shift the retention time and the mass. And then N-terminal cysteines, this is something we came across in an experiment once. When you alkylate cysteines prior to digestion, and then they're digested, if you have a cysteine on the N-terminus of your peptide, the alkyl side chain can actually react with the N-terminus, cyclize, and lose a mass, lose a proton. And when it does that, the mass shifts. And now if you're targeting that particular peptide, you won't see it because the mass shift is so large. So we try to stay away from those. And then N-terminal or C-terminal peptides within a protein. Sometimes if you don't have control over your sample prior to the point when you start having to process it, there are exoproteases that can be in the sample that tend to just chew the protein from the C-terminal N or the N-terminal N. And so we often recommend just not paying attention to the absolute N and C terminus and moving in several amino acids, like 5 or more, before looking for possible peptides. So for the targeted workflow, it's a bit simpler in my opinion, but I think it's also because I've just been doing it for a long time. So you start with your protein mixture, you digest, you add a heavy standard, and now you have peptides, desalt so that it's keeping your LCMS system clean, and then you run the LCMS gradient, extract the data. And here we've got a heavy standard spiked in. We look at the peak area of the heavy and the light. And when we know, when we look at those, we generate a peak area ratio. If we know how much heavy we spiked in, we can multiply this peak area ratio by the heavy amount, and that'll give us a value of in grams or moles or concentration in the sample. And so this is where we get the quantitative aspect. So when you're designing a targeted assay, these can be highly customized or not. So usually there's a good starting point, and then you can begin to play around with things like transition selection, collision energy, different digestion, and so on. So here are just some things that you can choose to spend your time on. So for those of you that are ASMS members, this is a poster that was presented in 2011. And so the particular details about the different digestion conditions we used are there. And if you need it, I can post it. But this was an experiment where we looked at 4 different digestion conditions, meaning we looked at different denaturation chemicals and different concentrations of denaturant during a lyse-C addition. So what we're looking at in this graph here is percent recovery on the x-axis, or sorry, the y-axis of peptides. And then on the x-axis are all the different peptides we were monitoring. And the different colored bars are showing the different digestion conditions. So the 2 molar urea and the 4 molar urea doesn't refer to the total urea that was used. We actually started with 6 molar urea and then reduced it to either 2 molar urea and added lyse or 4 molar urea and added lyse for a 2-hour digestion, then reduced the urea concentration further to less than 1 molar to do overnight trypsin digestion. So that was those two. And then the other two denaturation conditions were sodium deoxycholate is what the green DOC stands for. And then, sorry, that is an acid precipitated denaturant that once you digest, you can acidify your sample and spit it, and then precipitate from the deoxycholate comes out on the bottom. You remove your supernatant and technically your sample's clean. And then trifluoroethanol is shown in the purple. This is a volatile denaturant, so you could dry it after digestion, and this would theoretically all disappear and not cause any problems on your column, unlike urea, which you need to desalt before doing the analysis. So we looked at all the differences, and the differences weren't really that big. There were a couple that stood out. For example, the trifluoroethanol had much better recovery for these two peptides, but in general, the TFE was a little bit lower. And so, you kind of have to look at what's the best for most of the peptides. If you wanted to pull these two peptides out and process them separately, if that's an option, but this just gives you an idea of things to look for in general. So now picking your peptides and your gradient and your digestion, you don't necessarily have to keep them identical to what you used for discovery, because what's used for discovery is sort of like you're trying to figure out anything that's going on. And if you want an assay to be feasible, to make it to a clinic-like setting, then it needs to be fast, it needs to be inexpensive, it needs to be easy, it needs to be robust. So, changing digestion conditions and shortening gradients and moving to higher flows are all very good things to consider. So in looking at the spectral library and picking peptides, this is just an example of clicking on a peptide sequence, and I have a number of product ions coming from the +2 precursor. And this is what it looks like from the discovery data. So we see all of these product ions and they're labeled. Integrating this into Skyline is pretty easy, and there are tutorials on how to do it. So why do we do this? Well, sometimes when in the data analysis and you look at your list of peptides, you see that you're detecting the same peptide, but by more than one charge state. And so here's an example of a peptide where we see the +2 precursor on top and the +3 precursor on the bottom. And the question is, well, do you target both? Is that necessary? Do you have enough room in your targeted list? Or do you just want to pick one? And then how do you pick which one? And so what I do normally is I start with 2 and I target the 2 and I get the empirical data and I see which looks the best. But here there are things to consider. For example, the intensity for the +2 charge state only goes up to 160 e to the 3, so 160,000, but it's got a lot of product ions higher than the precursor, and that's usually preferred. Down here, the 3+ charge state it's at 4 e to the 6, so it's at least an order of magnitude higher than up here, but not as many product ions, and that Y6 is a lot lower here than it is up here. So these are just things to consider, and I would recommend just target them on the triple quad and then delete the one that looks bad. Sometimes peptides are beautiful. If you look at spectral libraries and you click on one, you see it and you're like, oh my gosh, look at all those peptides I get, those product ions I get to choose from. And then some are not. So here's an example of a peptide that was identified as something changing and we wanted to target it, but our options really are a B5 and a Y8 doubly charged product ion. We go down to the Y4, but there's a lot of noise down there. So this just didn't look that good. Sometimes even when they don't look good, I will set them up to target on the triple because triples can be more sensitive in certain ways than the high resolution accurate mass instruments, particularly if you're just looking for a few things. So for the best results, explore your data to understand it. Use predictions and algorithms as a guide, but it's always helpful to check the data manually because you might learn something. And then you build experience in generating targeted methods and you'll be able to catch more things and be confident in your data. And I like to think about this in like this old, story about a bunch of people blindfolded trying to touch an elephant and figure out what it is. And this guy has the ear, says it's a fan. This one has the tusk and says it's a spear. This one has a tail and says it's the rope. That's kind of what we're doing with the proteome. So, when we do this process, we can make assumptions, but not always what we see represents what's going on. So just a few more things. Refining transitions is an important thing to do, particularly if you want to target and not have to target lots and lots of fragment ions. I rely on something called the dot product and looking at the spectral library. And this paper tells you a little bit about the dot product, where it looks at the intensity and mass to charge of spectral libraries and of data, overlays them, and scores them between 0 and 1, and perfect is 1. And so here's some examples of looking at our discovery data from the high resolution accurate mass instrument, or maybe redoing it on the triple quad with similar conditions, and then trying to figure out which is the proper peak for this peptide. We've got this big one at 50.9, this not so big one at 35, and this little tiny one at 45. And if you zoom in, the one at 50.9, it's one transition really. There's not much there, and its dot product is 0.16, which is super low. The one at 35 seems like a good candidate, but when we zoom in, it also is really only one transition, and peptides don't normally fall apart that way. Its dot product is 0.37, so probably not that one. And the one down here at about 45 minutes, you see how it has a bunch of transitions that are like within 10% of each other is usually what we try to look for. And that looks a lot better. It seems like more pieces of that peptide are being detected, and its dot product is 0.92. And so this would show us that likely this one, even though it's not the most intense, is the peptide we're monitoring. Now, keeping that peptide as an example, we can also look at retention time from the gradient, which normally correlates with hydrophobicity of peptides. So here, that peak that I showed on the previous slide at 42.5 minutes with a dot product of 0.92 falls right on our regression line, where we have hydrophobicity score on the x-axis and measured retention time on the y. In Skyline, this is generated in Skyline, you can click on each of these dots and it will show you the peak. So that big tall peak at 50.9 minutes with the low dot product, it's a little bit further from the regression. And then the peak at 35.1 minutes is even more further from the regression. So not only does low dot product, but also retention time can help guide the selection of the proper peak. Here's another example where we've got this big peak that looks great, symmetry, it's got little bumps, but not a big deal. Or these other two peaks. This one has a dot product of 0.41, where this one down here at 44.9 has a dot product of 0.88. And it may be hard to see, but there are all of the transitions that are being monitored are lifting off the baseline. So they are all present, which to me would indicate it's likely this small peak at 44.9. So then I do my backup check. I look at the retention time, and this big tall peak, its retention time falls right on the regression, whereas this one falls a little bit further away. Sometimes that happens, and it's hard to know exactly which one is the proper peak. And I might try to keep everything in there until all the final conditions are met to see which one really falls through. But my guess was that it's this one at 44.9. Standards, why use them? Internal, external. Do you use peptide standards, extended peptide standards, protein standards, SILAC? So this is just a quick dive in there. So we use standards because mass spectrometry is really only semi-quantitative. All of the quantitation we do is relative to something else, whether it's relative to an internal standard, an external standard. Its peak area alone cannot give us a quantity of what's in the sample, mostly because of the background signal that's contributing. And we have this unknown coefficient that I'm just calling K in this equation that takes into account ionization plus a number of other things. So internal standards are the best ones because they co-elute with your sample. They act like your analyte, and then they can give you an idea what's going on and account for some of that variability in sample prep, auto sampler pickup, separation, electrospray stability, fragmentation, and detection. So internal standards versus external standards. The internals obviously can account for variability, particularly in the whole sample prep and LCMS workflow. External may not account for the LCMS variability as well, because they're being acquired at a different point in time. It may aid in interference issues, but the internal standards are usually better off for doing that. And so this is just a picture that shows you where you can add in your internal standards and what type. Heavy protein standards are great, but they're often hard to find or not stable or soluble. SILAC, you can label a global proteome if you're working in a cell line and use that as like a standard that you always compare mixed with another sample as an option. Extended peptide standards are peptides, tryptic peptides that are synthesized with the native amino acid sequence on the N and C terminus, so that when you add them to your sample, trypsin has to go in and cleave at the proper points, where it normally doesn't do when it's just a fully tryptic synthetic peptide. So those can be added in before digestion. And then regular synthetic peptides, and you can add them in a number of places here. So heavy protein standards are the best way to get closest accuracy without adding too much to your sample. You're not adding a whole proteome, you're just adding a protein. They can be grown on heavy media or with heavy arginine and lysine, but they may be difficult to produce or purchase or purify. And so these are all things to consider. And they're better suited for concentration analysis by amino acid analysis than peptides. And this just shows an effect of looking at an internal protein standard here on the right versus a fully tryptic synthetic peptide standard on the left. Here we're looking at, so we see an overlay of 3 peak traces. The brown is from the protein standard, the peptide that's coming from it. The blue is from the C13N15 labeled peptide standard, or the peptide that's coming from it. And then the red is the light peptide signal that's coming from the protein that was in the sample. And so in order to quantify, we generate a peak area ratio with always the light on top, and then we either divide it by the C13N13 peak area and 15 peak area and multiply by the spike level, or we can take that same red light peak area and divide it by the N15 protein internal standard peak area and its spike level. And this will give us a quantitative value of what was present in the sample. So this is the same exact data, just normalized to either the peptide from the C13N15 or the N15 protein peak area. And we can see that light peak area, which is the same from left to right, pops back up on the line when it's normalized by the protein standard. And that's because the digestion inefficiency when protein's being digested between the light and the heavy is going to be the same. And so if we add 25 femtomoles of that heavy peptide, we're not going to get 25 femtomoles of peptide back. We usually get less because the peptide may not digest to 100% efficiency. As long as it's reproducible, that's the most important point. And so this difference in looking at the amount of the light peak area by either the synthetic peptide or the heavy protein peptide usually is due to digestion inefficiency. So when we normalize with a standard that has to take that variability into account, we see that we're approaching 100% recovery, and we're right on that line. Okay, calibration curves are important. They are time consuming, but they're easy. So it's always worth your time, in my opinion. It will allow you to define the linear range, the limit of quantitation, the lower limit of detection, the upper limit of quantitation, and then it'll make sure that your response is linear, and it should be linear over your linear range. So this just goes through some ideas about what can affect the linearity. Start with the knowns. There are lots of ways to calculate your LODs and LOQs that you can look up. And space your concentrations wisely. If you do a concentration every order of magnitude, like 10, 100, 1000, there are going to be changes occurring over that range that you're not capturing because the points are too distant. So it's also important to figure out what level to spike your internal standard. Because if you have it spiked so high that it's 1000 times larger than your light peak area, that just is it's a stretch. So we always try to get the heavy peaks in line with the approximate concentration of the lights or within 10 or 20 fold. So we get the best measurement accuracy and precision. And so to do this, you can just inject your heavy peptide without the light into a similar complex matrix, and then see where its response is reproducible at its lowest, and look at any residual signal for the light peptide that may or may not be in the sample to see if that will cause any interference issues. For this group, probably maybe I don't have to go over all of the different choices of mass spectrometers, but I have a quick slide that just breaks it down into low resolving power, either a triple quad or an ion trap, or high resolving power like time of flight or FTICR and Orbitrap. And so these all have their pros and cons. And sometimes you don't get a choice of which instrument to use. Sometimes you are stuck with the triple, or sometimes you don't have one and you are stuck with a high res instrument. Either, all can do it. There are just things that you have to keep track of, like the triple quad is only capable of nominal mass, and the high resolution can give you accurate mass. So you can go through your data analysis with a little more confidence on the mass accuracy side over here. You don't need to do collision energy optimization for the high resolution accurate mass, but it's recommended on the triple quad because you can improve your gain and your signal to noise. But throughput wise, with the current state of the art, both instruments are capable of detecting at least 100 peptides per second without having to do retention time targeting. This briefly discusses instrument cycle time, which is the amount of time it takes to acquire all the data points in a given time frame. So this allows you to count the points across your peak, and you need to make sure you have at least 10 to 15 points across the chromatographic peak. You can have less than 10. 8 is fine, but when you get to 4, you no longer have a peak, you have a trapezoid, and that's not good for quantitation. So paying attention to that is important. I'm going to skip this for a minute. Last tips and tricks. So there can be duplicate peptides in your proteome, and Skyline helps remove them or allows you to monitor only one. So you're not analyzing the same data over and over, but it's important to make sure your peptide is unique. This is an example of interferences and figuring out if you have them by looking at the ratio of your product ions. So this is a full scan MS2 of this peptide. We see all the product ions labeled. And normally the ones that I was monitoring was this 430, the Y4, Y6, Y8. Y8 was always the tallest, Y6 was always the smallest, Y4 was always in the middle. And so by normalizing to the Y6 and making that peak have, that ratio be 1, I always got this ratio of 2.1 to 1 to 3.85, looking at these peak intensities. And that was true for the light peptide and the heavy peptide. So that was perfect, it matched. But when I started looking at these in real samples, like MOLT4 cell lines, I noticed that the heavy peptides tended to stick pretty close to that ratio, but the light peptides did not. What was happening is the Y6 ion became so super large from an interference that what I was quantifying was not just my peptide. And so I was coming with erroneously high quantitative values that I didn't realize until I started looking at the individual ion transitions. This is one way Skyline visualizes that. So these are contributions from 3 different transitions from a particular peptide shown in individual colors. These are replicates across the bottom. And that's the heavy peptide. The light peptide you can see down at this lower left-hand portion of the plot. This is like a low spike level, and that purple transition is huge. That's coming from the sample matrix. And as we spike more and more and more of the light peptide in, going to the right, you can see the contributions of the brown and the blue become bigger than the purple. But this is an important thing to pay attention to because this will give you false positive results. And finally, looking at some of the data, you can identify issues if you look at your response curve, because all of the points should really fall on the line. Here we're breaking things down and plotting the individual transitions. And we see the red one is like peeling off the curve, and that means there's an interference in that one transition. So the entire peptide doesn't have an interference, it's just that one transition. And then here, same thing with the blue. All the way on the upper right, we've got endogenous present in the sample. And we can tell that it's endogenous peptide because all 3 transitions are behaving the same exact way. So we know it has to be that peptide that's present at low levels in the sample. On the lower right, we have detector saturation. And we know this because the 2 most intense ions, these, I didn't tell you, these points are color coded. So red and blue and then green are the order of intensity of these product ions. The red and the blue, because they're the most intense, they're saturating the detector as they're going through, and so they become nonlinear at the top, whereas the green can keep going. And this one peptide, lower middle, was just not detected. And then on the lower left, we had unstable electrospray. So while there's this loose correlation going lower right to upper, lower left to upper right, the data quality is just super poor. All right, so do I have a couple of minutes, or should I stop?
Go ahead, Susan. You can.
Okay, because this is the cool part. So as we are all aware, in March 2020, the world began closing down because of COVID-19. Work was closed, schools were closed. Even today, my second grader son, someone in his class tested positive. So I'm home today and he's playing with army men over there. So you might see him if he comes through. So being frustrated because I can't do PCR and I'm not a biologist and I don't know genetics and viruses. I had no idea what to do, but I knew I had this large analytical lab at Northeastern University that needed to be tended to. I needed to make sure nothing broke during the pandemic and the shutdown. At the same time, you know, having 2 children in elementary school, remote learning at home, made me about ready to lose my mind. And so I was trying to figure out anything I could do as a scientist to contribute. So I found this paper from Ben Osborne, who's now a professor at Johns Hopkins, and he had published this pre-review online about predicting peptides, unique peptides from the SARS-CoV-2 virus. And so I reached out to him and I was like, Ben, I have a bunch of instruments and a lot of time. Like, how can I help? So he sent some samples and we designed an experiment to use a Q-Exactive and a Lumos with FAMES to do data dependent analysis and identify everything that was in the samples he was sending me. The samples he sent were a cell line that was transfected with a viral plasmid and then digested. So it was safe on my end, but we were just looking what viral peptide proteins are being generated. And then the goal would be I would take all this information and set up a targeted workflow on the Altis, going incrementally from what I normally do to making it as fast as I possibly can. So this refinement process here was we were able to detect 395 theoretical peptides from the discovery experiments, and 14 of them. So that was like the theoretical, we didn't find those, sorry, those were the predicted. We found 14 of these peptides in the HRAM data, and they were primarily from the nucleocapsid, which is shown in here, the little red guys. So I put 23 on the list for targeted selected reaction monitoring, just to see what else in the protein we could detect that maybe we just didn't get from the triple quad, or sorry, the high res analysis. And the best 7 made the final cuts. And these were based on stability of the peptide, peak shape, and so on. So I started with a 55-minute nano LC method and then was able to shrink it down to a 15-minute final method. And at this point, heavy peptides from JPT were commercially available. And so I spiked them in. So this is showing you an example of the 15-minute method. This is our pressure trace. And so I realized after going down to the 15 minutes, I'm like, oh, I've got all this column equilibration time. I could shut this down earlier. And then I could also steepen the gradient and move the peptides a little bit more to just end it at 9 minutes. And so ultimately, that's what I wound up doing. But I'm just going to show you the 15-minute method compared to the 50-minute method here. And we see the series of peptides and their peaks intensities are on the same scale. The chromatographic time obviously isn't, but going from a 15-minute method to a 15-minute method, 50 to 15 worked really well. So that was great. I was never able to get my hands on real human samples to test. Ultimately, I think it will be difficult because they're often stored in viral transport media, which contains fetal bovine serum. So now you're fighting against a bovine proteome and a lot of bovine albumin, which may swamp out the sample. So others are coming up with different ways to do this. And there is an anti-peptide antibody method that will capture these peptides out of the viral transport media. So I'm glad to see others were thinking like me and were more successful too. So will it work all the time, even with lots of hard work? And unfortunately, the answer is no. But we often have a mantra, and people who've like worked with me know that I have a rule: there's no crying in mass spectrometry. So yeah, can I say I've lived by that rule? No, but there are frustrations and things, but they all can usually be overcome. And, you know, reaching out to people and networking and finding out what worked for them is a great thing to do. So in summary, I always recommend consult the literature, generate the data, review the data, see if the data makes sense, and then repeat. So with that, I'm happy to answer any questions. Thank you for your time, for your attention. It's hard to know when you're doing this online if people are like enjoying it or not. So I hope you did. And yes, thank you.
Thank you, Susan, for that beautiful talk with a lot of experimental advices, which is something that we always need. And I'm speaking for me, but I really enjoy your talk. And while we wait for the question from the audience, I would like to ask you a few things. Well, of course, with experimental things, given your expertise. You talk about gradient length and flow rate. What about column length? Have you got experience on column length?
Yes, so there are a variety of things to consider with columns. So column length or particle size are two important things that are linked. So what it comes down to is the number of theoretical plates you have in your column. And the two ways you can increase those, which improves your resolution, is by lengthening your column and having more beads, more theoretical plates, or by shrinking the beads and making them smaller. So in the same length column, you just have many, many more smaller particles. And this was something that came up a few years ago when a lot of people use Reprosil C18 from Dr. Meisch in Germany, and those are 3 micron beads. But then when a 1.9 micron bead diameter was made, many people moved to that, packing the same length, but getting tighter, sharper peaks. The downside is whether you go to a smaller particle size or a longer column, the pressure. And so some of the columns you're kind of limited to using UPLC type systems because the pressure becomes so high that you exceed like the 400 bar or whatever the limit is on your LC. There is also, I don't know if you've seen the it's UPAC. It's these columns that are, they're made with like pillars, and they're very, very low pressure. And those,
monolithic.
Yes, the monolithic. And so I've tried those, and the pressure is extraordinarily low. And the separation is online with the C18 separations that we normally see at much high pressure. So I'd be interested to see where that technology goes, if they can increase the number of the pillars, or like the flow length, or the distance, decrease the distance between them, if that would improve resolution without driving up the pressure of the system.
Thanks. And you talk a lot about desalting. Do you desalt offline, and do you use a trap column simultaneously, or one or the other? What do you advise on?
Yes, I do a variety of things. So for the peptides that I was analyzing from the nucleocapsid from the virus, I was not desalting them because I wanted to try to produce a method that was robust and easy and didn't require a lot of steps. So if it were ever able to go into a clinic or a testing lab, it would be fast. So for that, I had a trapping column, and I took the sample and I loaded at a higher flow rate onto the trapping column and then switched the valve, ran the gradient over the trap and the analytical column. The analytical column was a short 5 centimeter, 1.9 micron beads to keep the peaks sharp and narrow. But I didn't want to have to deal with offline desalting. Now, for discovery proteomics and trying to detect the variety of everything you want to detect, I desalt offline and I don't use a trap column. Now, the limitations are a little frustrating because if you load a microliter at 300 nanoliters per minute, it takes 3 minutes to load. If you load 5 microliters at 300 nanoliters per minute, it takes 15 minutes to load. And that's a very long time, in my opinion. So I don't love not having a trap column, but personally, I've done like my own in-house evaluation of loading with the trap at different flow rates and for different lengths of time. And you can lose hydrophilic peptides and you can lose some early eluders, and the peptide IDs, the numbers tend to be lower when I've used a trap column. So when I have people coming in and they want like sensitive proteomics experiments, I will desalt offline.
Thank you. And one final question for me. As mass spectrometers become more and more sensitive, what is your approach to carry over between samples? How many blanks do you run between samples, or if any at all?
That is a great question. And in running the core facility, it was interesting. A lot of people didn't really want to run either blanks or replicates because of it cost them money to run, you know, on an hourly basis. So I would make sure that the cost of running blanks was like, we're going to do it. And it wasn't an incremental thing. So yes, running blanks is important. And I mean, the cheeky answer is keep running blanks until you don't see carryover, but that's not practical. So the ways that you can get around that is either a separate blank injection where you run what's called a sawtooth gradient, and you go from low organic to high organic very rapidly, like over 2 or 3 minutes, and then drop it down very fast. And so you wind up having something that's slanted like a sideways triangle. And then you do that multiple times in a short period of time. And you can often inject maybe either organic solvent or I sometimes will inject like my peptide standards on that. And the reason is because I often see when you're doing this kind of blank injection, if you just inject mobile phase A, then really only the gradients are going over the column and there isn't much interacting with the stationary phase. Whereas if you inject something that will go in and equilibrate between the stationary and the mobile phase, like, I don't know if this is true, but I feel like it helps the stationary phase kind of move around more and do what it's supposed to, and perhaps allow some of the carryover to be released. So if you do a separate injection that's a sawtooth, that's one way, or you could have them included at the end of your gradient before the next injection. But I always will run a blank before the next injection to ensure that there's not carryover, and those approaches seem to help me.
Thank you. Soledad, I don't know if you picked up a question from the audience.
Okay, I'm reading here on YouTube. I think we don't have a more question for Susan. Just one from me. It's like I feel that you gave us a very complete presentations about the tricks and the tips to deal with the analysis of. And I would like to ask you, how do you control, or how much variability you allow to your system in terms of biological variability? No?
So first, one thing I didn't mention was I always incorporate a system suitability evaluation in every and any study I do. And it's usually I will run it before a study, during a study, and after a study. And I monitor the variability of my analytical instrumentation that way to make sure that it's not exceeding 15%, or, you know, my retention times aren't shifting like crazy. For variability within a sample, for discovery proteomics, it's always a bit higher than for targeted, but you try to aim for as low as possible. And like 15% is usually, I don't feel so comfortable going above that, but for discovery, 20% or 30% variability within a sample. So this is like replicate measurements is fine, just due to the sampling nature of the instrument. However, the biological variability that you can measure has to be greater than your analytical variability. So this is the issue where if your analytical variability is 30% and your biological variability that you are measuring, you think is 30%, is it really? And so that's, we just try to do as much as we can with the instrumentation and, you know, making sure that we preemptively, I didn't used to change my solvents as frequently as I do now, and I didn't used to run system suitability as frequently as I do. And there are a lot of little like routines that I put the LC through, kind of like as you get older, when you get up in the morning, you can't pop right out of bed anymore. You have to like stretch a little bit before you get up. So that's what I do with my instrumentation to help keep them operating very well. And so that will keep the variability of their operation lower so that we can begin to see smaller biological changes occurring between samples.
Thank you, Susan. And thank you also, Germán, for moderating the talk. People here on YouTube are also thanking your presentation, and it has been great and a very generous way to give us a lot of tools to deal with the proteomics analysis. So,
sorry. Thank you, Susan, for me as well. And just a comment from the YouTube chat. A great presentation, and we need a journal of negative results. And in mass spectrometry and in proteomics, it's really important what you said, that many things that go wrong never gets published. And in a comment for me is that you learn a lot from social media, and I encourage you to follow Susan on Twitter. You mentioned Ben Osborne, who has a great blog on proteomics, and please, Twitter and the mass spectrometry community, the team mass spec, follow all of them in Twitter, and you learn a lot.
Thank you. We have that opportunity now. We have these tools of from social media, and we can use it for also enjoy and complete our understanding of, well, the negative and positive results. No? That's a good way. Well, finally, thank you so much for the presentation again. It has been a pleasure to have you today for this cycle of talks, and we are going to have the third one next week with Dr. Omar Amador Muñoz. Thank you all for being here, and see you next week. Thank you.
Thank you.
Bye.
Have a good night. Thank you.
Thank you. You too. Bye-bye.
See you.